all right. Apparently, we are live, and I'm back doing the show. How? Let me just check to see if everything is running smoothly here, guys. Just give me 1 second. Apparently, we are good. Good. All right. So, welcome everybody. It's with great pleasure that I have Dr. Melissa Oddie again here with us tonight. Let me bring her on. All right, Melissa, how are you?
I'm very good. Hello, Humberto, and hello, everyone.
Yes, it's great to have you back here. So the idea tonight is that since our last live stream, you and the videos that I made about your work, I got a lot of attention, and a lot of my audience like really enjoy your approach, your discoveries, the way you talk and teach, and you got a lot of attention. A lot of people request me your phone number, email, and all the things. They want to do projects with you, and so wow, that's great. So there is a lot of attention in that direction. I noticed too,
That's
meaning
fantastic.
beekeepers are looking for alternatives, and they try to find ways to improve their bees. And what you brought to the table apparently is something that people are looking for. So why not bring you again, since you have a very interesting project that I want to talk about later on in this discussion, in this live stream, which is this book, because this
Yeah.
match this whole thing that I'm talking about here. Because after I don't know how many years now working with that, with hygienic bees, and discovering this new behaviors, or sort of new, you know how that works.
Adapted. Yeah.
Yes, adapted behavior. So when you put it in a book, people can absorb that in a more organized way, instead of getting things one a piece of the internet and linking things together, things like that. So
Yeah.
that's what I want to do tonight. I want to go over, if we can, to that paper, the 2021 article of yours, so we can
Yeah.
go a little bit and talk about that, the discovery. And then I want to hear from you, since that publication, what happened with your life?
Yeah.
We know so many different things
Lots of
might have
things.
happened, and to that end up with the idea of publishing a book about it. So can
Yeah.
we, shall we?
Yeah. Let's do it. Let's start with the paper, and then we can transition into talking about the book and all the stuff I've learned since I've started.
But before we start, I want to say one thing first. I want to say hi to everybody at home. So let me see. Apparently, the chat is working. Wildwood Honey Farms. Hello. Hello, Marcus. Hello, Line. Jason. Lean. I probably messed up your name. I'm a master to messing up people's names, so don't be offended. Um, so okay, it's working. Everything's working. But one thing I want to do too, it is to say thanks. Wait a second. I need to share my screen too. Okay. So before we go to this article that Dr. Audie we're going to talk about, I want to say thanks to our sponsor tonight, which is Apis Tactical. I don't know if you guys saw this company, but they're being very interesting approach about better gear, better for beekeeping. I talked with the CEO a long time ago, and he had this idea to bring a little bit more fashion to the beekeeping industry, and I truly understand that. Not only they design a glove that really fit really good, I like the glove very much. It fit my hand pretty good. I can use my fingers to get the frames. I can be more precise with those. But on top of that, they're looking good. You know, sometimes in a beekeeping world, the way we dressed ourselves is kind of embarrassing, to be honest. And but those gloves and the projects he has coming up is pretty nice. So stay tuned. Apis Tactical is bringing some new gear for us, protective gear for beekeeping, and it's really good to have them sponsoring the channel and allowing me to keep moving forward. For example, tonight, bringing Melissa back. So Apis Tactical, thank you very much. If you guys at home have want to check them out, if you check them out, you are helping the sponsors, you're helping me. All right, thank you. Melissa, let's do this.
All right.
I need to remove this. Boom. And where are you? Here we go.
There we go. I need to ask you, do I have control of the slides, or do you have control of the
You
slides?
control the slides. So the floor is yours.
All right. So this was a paper that was kind of like an addition to the 2018 paper that we were looking at. So basically take a bit of a deeper look into recapping and how it's associated with the surviving populations that we were looking at. So I'll give you a quick rundown of that paper, and we can talk about what that means and how maybe beekeepers can use this information in their yards. Oh, good, it works. Nice. Okay, so I'll talk a little bit about Varroa for the new timers or the newcomers here who don't know a lot about it or who are just starting beekeeping. Why Varroa is bad, and you know, and what we're doing to try and fight it. I'll talk a little bit about how I started in bee research, my work in Norway with a resistant buckfast population, and then I'll talk a bit about the paper, what we were doing, and talk about this behavioral trait recapping and its association with brood removal and Varroa resistance. So to start, Varroa is an invasive ectoparasite that feeds off of honeybees. It feeds off the fat body tissue, and it invades developing honeybee cells and bites the larva, and it can transmit pretty damaging diseases. And this might, because it's invasive, our Western honeybees don't have a lot of defenses against it, so the population growth of this parasite can be exponential. And that, coupled with the viruses, can kind of kill off colonies very quickly, depending on the density of colonies in the area and the level of threat of the mite. Um, it can adapt to a lot of chemicals if chemicals are used very consistently over a long period of time. They can develop resistances and have developed resistances. So a lot of beekeepers are looking for more sustainable alternatives, and some of them have turned to evolution as a way to solve the problem. So adapting their bees to overcome the parasites so that they can kind of keep level and just kind of keep working at stopping the parasite, no matter how the parasite would change. So we actually know of several populations that have the ability to do this now. There are several populations in Europe. We actually have new populations, which we're going to talk a little bit about in the talk after the paper. You know, we have evidence now of populations in Cuba. We knew about the South American ones that have Africanized genes, but the Cuba population actually is fully European, almost exclusively. You know, we have a number of them that are resistant all over the world. So we've got a kind of a large pool of resistant colonies now that we can start to learn exactly the best methods for selecting, and you know what we can do in other places around the world, how we can use that information. So I'll start with what I've been doing. So Tarja Reinertsen was a beekeeper in Norway, and he had basically stopped treating his bees in 1998. The mite arrived in Norway in 1993, and he lost a considerable amount of bees to the mite. They started treatment regimes fairly quickly afterwards, but the treatments ended up being a little severe for the Norwegian climate because the levels, I guess, were set to further south in Europe. So a lot of colonies were still dying off. So Tade decided to stop treatments and then work with the bees without treatments. And he ended up, you know, losing a lot of bees in the first few years and struggling with it. But eventually he managed to breed back his stock, and he got you know, commercially competitive honey producing stock of buckfast bees that were completely unaffected by Varroa. They still had Varroa, but they were able to survive just fine with low levels of the parasite consistently in the hives. So that's where I came in about 2014, 2015. I was asked to go and visit Norway to meet with Tarja and study his bees to see exactly what was going on. So I took a number of a number of years of data, and this is the bottom board counts that I was taking in various given years, and the level of mites on the bottom boards, this was kind of a daily drop sort of situation. Tadia's colonies are all in blue, and the controls that I was taking round about the same area from colonies that were being regularly treated with formic and oxalic acids. Um, so right away you can see that the Tadia's colonies had much lower mite levels. No matter what time of year that I was actually taking the data, they had consistently lower mite levels, which was really interesting to see. So like throughout the season, they could keep the mite levels low. So a little more in-depth investigation to that. I looked at, hang on, we are missing a line here. That's very interesting. I guess it didn't translate very well. So there's supposed to be a blue line on this graph. I'm going to have to describe it to you. When I basically took bottom board counts for an entire season, so every week I would pull out the bottom board count and then put it back with fresh paper on it. So I got a consistent increase in the mite levels. So here you can only see the susceptible colonies, and you can see the exponential growth curve that I talked about. So the mite levels just increased. So you're going to have to imagine the blue line, so Tarja's bees. It started at 5, so same place, and then just kind of kept going at 5. And I think it ended up to an averaging about 8 per day in mid-August, which seems very high, but the bees pretty much survived relatively well, and they keep surviving. So the line statistically did not move from 5. They weren't statistically different between the beginning point and the end point of that blue line. So you're going to have to imagine that it's there. I'm not sure why it didn't translate into the presentation, but so what that basically means is this resistant buckfast population basically kept the mites at the same level for the entire season. There was no point where the apiary that I tracked ever got out of hand. Whereas you can say I actually had to stop the experiment early for the control colonies because we were just going to lose them. There were so many mites in them. So why did that happen? I did a study. This is before the 2018 paper. This was when I actually was looking during my PhD. One of the first studies that I actually did. So to investigate hygienic behaviors, what I did was I went to a group of colonies, control colonies that had nothing to do with the control group I was studying at the time, and also Tarja's buckfast bees. I went to an apiary that we knew had high mite falls. And what we did was we caged queens and got donor frames of brood, waited till they were capped, and then I photographed and mapped them and then transferred them. Half of the frames went to Tarja's buckfast colonies and half of the frames went to the control colonies. And I kept them in these colonies for a period of about 9 days. And what that did was basically giving workers of each colony type access to this brood to see if they were going to do any sort of removal or hygienic behavior on this brood. And then after the 9 days, when the Varroa had sufficient time to reproduce in the cells, I removed them and froze them. And then dissected the cells one by one and counted all the little mite families. So I got kind of an average reproductive rate of the mites. And what I found was really interesting because the brood came from the same place. And the only difference was that they had 9 days in Tarja's colonies or 9 days in the control colonies. And immediately what I saw was that the brood that had been given to Tarja's colonies had a lower reproductive success rate than the brood that was given to the controls. And again, this brood was from the same place. So it means that something that Tarja's bees were doing was reducing the reproductive success. Now, interestingly, it's not shown here, but I did actually look for brood removal because I did photograph and map the frames after they came out of the colonies. And then dissecting them, I got to see whether there was brood that had been relaid and just was not age appropriate for the study. And there was no significant brood removal difference between these two colonies. So the Tarja's bees were reducing the reproductive success, but they weren't removing any of the brood. So this is the mystery that I was given. And interesting, what they were doing was they were doing something called cell recapping. Now, what this is, it was described by the Baton Rouge lab way back when by Harrison and Harbo as kind of a byproduct of brood removal. So it was an investigation behavior that the bees perform. They open a cell just so they can see what's going on inside. If the brood is sick or damaged or injured in some way, then they usually just uncap the cell completely and remove the brood. So this was a behavior that was it's pretty much in all honeybee colonies as a general hygienic behavior against disease. But what I saw was that the recapping behavior was much, much higher in Tarria's colonies. So they didn't seem to be removing more brood than the controls, but they were recapping at a much higher rate. So I thought, this is definitely worth investigating. So after that, this is when we got the data for the 2018 paper, when we went to our colleagues in France and Sweden. We said, hey, can you just go out into your colonies and look for this behavior? And so we did, and that was the result of the 2018 paper, when we found that pretty much all of the resistant populations that we had and were investigating, all of them had higher rates of recapping, regardless of how genetically connected they were. Most of them weren't even related at all. So you can see this graph might be a little small, but all of the black bars belong to surviving colonies, and then the dotted lines divide each of the populations. We had 2 in France, 1 in Sweden, and 1 in Norway, and then the light gray bars are the control colonies, and the x-axis is the different countries, yep, and then the y-axis is the level of recapping. The 2 different bars per population are cells that we found mites in, so cells that were infested, and cells that were uninfested. So the highest bars were in the surviving colonies and in the cells that had mites in already. So the recapping behavior was targeted towards the Varroa. So that's what we found out, and other scientists have now kind of repeated these results and looked for recapping behavior in other populations. And I think pretty much in every population of resistant colonies that we know of, we have found it where we've looked for it. So it's in South Africa, it's in Brazil, it's in Cuba, which is really cool because it means that this is probably highly associated with surviving bees with Varroa resistance. So, my next question is, is this a reliable trait? Can we use this somehow? So there's two different elements to this trait that I was investigating when I was doing this type of research. There's 2 elements to recapping. One is the frequency at which the behavior is performed, and the other is the accuracy. So how often they actually get an infested cell with this behavior. And these 2 different elements of the trait can vary independently. So you have some colonies that do a lot of recapping, but they're not very good at getting the infested cells with this behavior. And then you have colonies that maybe don't do a lot of this behavior, but are very accurate when they actually do. And I found that colonies just across the spectrum, some colonies are great at both, some colonies are only great at one or the other. But the most successful colonies were good at both. So now here's the difficult part. So in addition to these 2 different elements of the trait, you also have environmental variation. And that is, the bees are not going to expend effort when effort is not needed. And that's usually kind of the rule of pretty much all of the animal kingdom. So if you have a situation where you have a lot of mites, they might switch on and you might actually get more of the behavior. You might observe more. They might even have a higher accuracy rate. But if there isn't a lot of threat, so there's not a lot of the trigger, then the bees might actually just ignore the few mites that there are in the cells and not perform the behavior. Which is, it's fine. Energy conservation. This is great for bees because they're not going to spend effort that they don't need to spend. But when you're looking for the trait and you're looking to quantify the traits and make assessments based on the trait, if the mites are under control, you're not going to see a lot of this performed. So you might then underestimate the potential of your colonies for survival if they don't have enough of a trigger to actually see this and quantify this behavior. So therein lies the problem for using cell recapping exclusively as a selection tool. But we're going into the paper now. So there was one thing we didn't do in the 2018 paper, which I really wanted to, is I wanted to find, I was looking for evidence that recapping was kind of directly involved in Varroa survivability, because we had this association with high levels of recapping. But because of the selection bias introduced by the bees, we couldn't really assess whether or not recapping was actually affecting the mite reproductive success, because the bees would, ignore cells that had a low trigger value. So if the mite wasn't reproducing, the bees might ignore it. So therefore, it introduces a bias when you're actually trying to calculate exactly how much of an effect recapping has on Varroa offspring and stuff like that. So what I did was I took all of the colonies that we had close to 100 colonies in the data set, and I plotted the colonies based on the reproductive success of the mites in that colony as an average, and the recapping accuracy. So how frequently the bees recapped cells that were infested with Varroa. And we had all of the control populations. So that was the control populations in France, Norway, and Sweden. And then we had all of these surviving colonies in each of these country groups. So it looks a little bit like confetti, but what we did actually pull was a slight correlation with, the more recapping, or the more accurate the recapping is, the lower the reproductive success of the mites is. And the pattern was statistically significant. Which is, it's good. It's not a ridiculously strong evidence, but it is a correlation, and it's a pretty strong one. So we had evidence then that, the colonies that were doing better recapping did have lower mite reproductive success. But again, I'll throw into that the environmental variation. The reason why we're probably seeing this vast distribution, and the control colonies are all over the place anyway, was probably because we were getting these colonies at varying degrees of threat level. So colonies that had lower threat level might not have actually done a lot of recapping. And what this graph does not actually display is how many cells in each colony were dissected, how many infested cells were found. And I guarantee you there's some colonies that had maybe 5 to 10 cells, while some colonies had over 30 cells. So this particular variation, we're still working on trying to get under control, so that we can have a valuable estimate, so a reliable estimate. So kind of standardizing the presentation of this trait somehow. We're still working on that. So this was the central graph of the paper. And the other one is basically, we took an average of the recapping accuracy, and then we plotted how many colonies in each group were above or below this average. And we found that pretty much most of the colonies that were above the average of recapping accuracy of 30% were in the surviving group. So colonies that had been selected or left alone to deal with the mite themselves, they pretty much always had the recapping accuracy higher than the controls. And so that's basically the gist of the paper. And we can go into questions after that. I can just sum it up. So, recapping is always higher on average in colonies that have either been left to their own devices to deal with Varroa, or have been actively selected to deal with Varroa. A single snapshot like we were taking, we're just taking brood samples and then analyzing those brood samples. It reveals a very high level of trait variation that introduces a lot of bias to the data. So you can't really get a good picture on exactly how much of this trait is expressed by colonies. So the maximum expression point of these colonies, you can't find that with just this snapshot, because some colonies are very highly triggered with mites, either after a robbing event or because they live closer to other colonies. Some colonies aren't triggered because they've handled the mite and they don't need to actually perform this behavior. So using recapping, trying to measure it directly and using that as a grading tool is probably not going to be that effective. But at the same time, looking for recapping in your colonies, seeing the caps removed off of developing brood, is probably a really good sign that you've got a colony that's switched on to Varroa and what it's doing, And that might be a better colony to select for resistance or to maybe put through more rigorous testing. So I would say selection is better done on performance-based traits. So how well they're doing with the mites and how few mites there are, and how little mite population. So the lower mite population basically are the colonies you want to be focusing on in the end. And that's basically the gist of it.
Fantastic. We got a fantastic overview about the whole thing. And I can see in the chat that people have a lot of questions. They were holding on to some questions. So that's why you're here. My
Yeah.
people want to consume your knowledge. So let me put this here. I'm gonna start with a good friend of mine. We never met personally, but one day that's gonna happen. My dear friend, the Canadian Beekeepers blog,
Hey,
famous guy,
Canada.
great, a lot of big good content coming from him. I learned from this guy a lot. Just watching his videos when I was in my transition, adapting from a different climate here in US, coming from Brazil. A lot of my knowledge from how to deal with bees and cold come from this gentleman. I appreciate his effort to bring content to the internet. So I'm gonna start with you.
Yeah.
So he do you know the mechanisms that kills the mites when the bees open the things? I think that's the question. What exactly happens with the mites when the bees open the cap? So why that reduce the reproductive success? Do you have any idea?
Right. I love this question because basically what we're trying to do is trying to get around the selection bias introduced by the bees, because they know what they're recapping. They know what works and what doesn't. We don't know. And a single snapshot, like I explained, is probably not a good representation of exactly. But I've got some theories and I can share them with you. There was a paper published by the Baton Rouge lab by Harris and Harbo, again at the beginning, that looked at what could be happening in these cells outside of the removal. One is, so the mite is generally very sensitive to temperature variations, humidity variations, and if the founders is sensitive, the offspring is even more sensitive. They don't have the chitinous protection. They dry out very quickly. So the reason why they reproduce in this cell is because it's this very homeostatic environment that keeps it its absolute perfect temperature and humidity. But if you were to disturb that, like say tearing open the cell and exposing that cell to open colony air, it could create a lot of problems for the reproductive process of the mite. So the reproductive process, basically how they start is they start laying eggs, and they lay a male egg first, and it's close to the top of the cell, out of the way of the shifting pupa, because the pupa basically sheds a skin in order to start the metamorphosis. And mites can get trapped and crushed by the pupa, so they lay the egg closer to the top. But at the same time, it's vulnerable if the cell cap is ever opened. So it could be that some of the eggs are getting damaged or destroyed or just drying out if the cell is opened like that. And also, offspring that have already hatched, they're also more susceptible to drier air and disturbances. They might be killed by bees looking at the cell and then taking the cap off and basically investigating. The other thing they could be hindering by taking the cap off, if they could be hindering mating success. So the varroa mites, they mate inside the cell, and if the cell is disturbed and opened, it might actually disturb the mites and reduce the mating success and sperm transfer. And Harrison Harbour in the Baton Rouge lab, way back when, I think it was 1999, they actually did look at sperm count in Varroa that had been disturbed by recapping and stuff. And they actually had, in colonies that were selected for Varroa survivability, for Varroa resistance, they had lower sperm counts. So it could also be hindering mating as well. And of course, if you remove the cell early, if you do actual brood removal, that definitely hinders reproduction as well, and it stops the first reproductive cycle, and it might prevent the new foundresses from actually being mated properly. So so that's some of the things that could happen in recapping, and we're still working on studies that will test that. So I've been working on a study that basically where I take a colony that is regularly treated, so it doesn't have very high levels of recapping, and I've been recapping the cells myself. It's very difficult because the bees kind of cotton on to what I'm doing, and they end up removing most of the cells. So I need to perfect this method so I can actually get a proper paper out with strong statistical results. But from what I was initially doing, it looked like that the most of the cells actually had fewer males, but I can't confirm that statistically yet. So that's all going to be theoretical until I can actually get a paper out on it.
Interesting.
But those are my thoughts.
Oh, yeah, you brought a lot of information for our to process. And I want to add a couple things there because I spend a lot of part of my life doing exactly what you're seeing in the screen right now, trying to make these guys to reproduce without bees or in a laboratory conditions. I follow many laboratories doing that. And it is a mystery to me why we cannot get rid of these guys, because they're so sensitive. They die so fast with anything. They're so fragile. And why they're not so fragile inside the hive, to me, is a mystery. But anything we do at the lab, it's too much light or too much water, or less water. It is
Yeah,
so.
I think we've only recently actually found a way to make them reproduce outside of a colony, just
Yeah.
inside cells, but outside of a colony.
But even though they depend on some feed, that there is some things that comes to bees. It's not completely, it's not 100% without bees or bees juices or stuff like that. And the ratio they reproduce is not a big of a deal. It's not much. We're not able to do much scientifically with them, unless I don't know what you're talking about and there is something new that I don't know, which is always great. If somebody figure that out, that's going to be a breakthrough. So, but it's so sensitive, and I always take my sleep at night to think about why they're so wimp. Why they? And that tells me a lot about the bees, because if they're so wimp outside the hive and so sensitive, they need specific cues all the time to triggers everything they need inside their physiology and biology to reproduce correctly. The bees must do a fantastic job keeping the environment perfect, for not only for the mites, of course, but for their own bees to raise bees. So again, the bees are fantastic, and because the bees are fantastic, a guy like this take advantage of them. And so this is a little capsule of pill that we put a larvae in it, and then we try to see if they can reproduce inside. There's a lot that can be done scientifically with these things, but it's fun to do it. But when you don't get the results, it's welcome to research. 99% is fail. All right, so let's see if we can get more things here. Uh, it's true, my friend. It's true. Don't need to be humble. It's true. One day we're gonna shake hands. I'm looking for that day. What else we have here? It's time to make questions, guys, because I'm really bad at this. I need an assistant. I need to bring somebody here and help me with
You want
this
to
thing.
maybe scroll up? I think I saw some questions
If you
further
say,
up.
if
There's
you
definitely
see
one at the
some,
bottom.
the bottom, okay.
Yeah, from another question from Canadian Beekeepers blog. Should I just read it out and say?
Is this one?
That's it. Yeah.
Here you go.
So do you know what stage of brood is most likely targeted for uncapping? Yeah, we do. So in the studies that I've done, the target was most likely at stage maybe about it's about 7 days post capping. So the mite starts to lay just after pupation or just during pupation. So the brood is in there for a few days beforehand. So it's pretty early that they start recapping. So when round about when the mite starts to reproduce, the mite starts to lay eggs, that's when they start uncapping.
Interesting.
and they might actually uncap a cell more than one time. I don't have video recordings of this behavior, but that's going to be one of the next studies. Hopefully, is to put these things in an observation hive, and hopefully we won't kill all the mites doing that. But just film them and see how many times they uncap. I know. I think Ralph Buchler may have some video footage, and I think he might be writing some stuff about that as well. So keep an eye on Germany and what they're doing.
Oh yeah, keep me in the loop on that. I would love to bring these people in so we can discuss and get
Oh
some
yeah.
footage from them, and you can spread the work these people are doing, so we can spread the word. Spread the word. Melissa, talking about that, what do you know other people what they're doing, results they're getting, something unusual, something different that we should share here with the people that cross, you've been doing this for so many years.
Yeah, and I think the one striking thing that has come is while we've been working with this and talking to beekeepers and stuff, there have been a lot of beekeepers from all over the world that have actually come forward saying, oh yeah, actually I don't treat, and I've got resistant bees, and we don't use any sort of chemicals on our bees, and they seem to survive just fine. And these are cases not just in the northern hemisphere, but pretty much everywhere. Everywhere you've got European bees, everywhere people are keeping bees or breeding bees, you have some cases and some levels of resistance. So it's a very common phenomenon, and we're learning about it. So when I started my PhD, and I was speaking to some of the beekeepers, I made a prediction. I was like, there is definitely a lot more resistance going on than we know about scientifically. And it looks like I was right. There, most of the beekeepers that came forward actually they do have some level of resistance in their hives. The interesting thing though is trying to move the bees out of their own specific environment tends to reduce the effectiveness of the survivability. So I think if I had a recommendation, I would say really looking for local stock or breeding your own bees to be resistant to Varroa is probably the best way to go. And importing is secondary. That's definitely the advice I'd give to Australian beekeepers who I know have recently got the mite and it's been spreading. I think it was just found in Queensland recently. Is definitely work on breeding from your own stock, and really do not try to import resistant bees because your bees do have what they need.
That's well said. I would say the same for my Australian friends. Just it's something new coming, and you're gonna need to adapt yourself too. Just learn everything you can possibly learn with other folks around the world. But at the end of the day, you're gonna have to create your own protocols based on your location and environment and plants around you and other pests. Too many variables. Welcome
So
to
many,
beekeeping. Yep.
so many.
Yep. So, hey, James Lee Bees, I know you're talking a lot of with the folks there, and I know you have a lot of experience. Do you have any questions? Just bring into the chat a little bit of your experience, James. And I'm looking
Yeah, you
forward
can
to see
definitely
you in
like
Michigan too.
re-ask your question. So put it at the bottom of the chat so we can see
Yes,
it
yes. If you have questions, please rewrite them because it's
if we didn't
easier.
get you.
It's easier. So while they're finding their questions, Melissa, let's talk about the book
Oh yeah.
because let's talk about the book
And that's
because
a good segue, when I was talking about all of these different cases that are coming
yeah,
forward, me finding out about stuff and being invited to talk with beekeepers. So that's the kind of that is the principle of the book. We have so many examples now and really well documented examples. Um, so I thought basically, and I met other researchers who are working on this. Stephen Martin is a really big one who was looking at. He's been researching Varroa resistance for over 40 years, and Marla Spivak also. So these are the two researchers that actually agreed to be the co-authors on this book. So we've been working together. The Kickstarter is my brainchild. So I thought I'd try and pull in a little bit of funding if we could, so that I could focus primarily on writing this book, doing research for the book, going out and reaching out to these beekeepers who have these resistant bees all over the world, and learning what they do, kind of writing down their methods and writing down their stories in immense detail, and then taking all of this data, everyone, and drawing parallels between the different cases so that we can come up with kind of a best practice recipe for people who want to start breeding resistant bees. And as far as I know, this is the first book that is attempting to do this, to actually look for all of these cases and present them in a book where everyone can see them in black and white, and then take the best methods from each of these cases and kind of give advice, basically from this pool of many. So that's basically the principle. That's what we're trying to do.
So let me see
And
if I got it, because when I knew about the project, in my mind I was kind of have some questions like how where the idea come from. I know you got an idea, you were working with this for many years now, and you have a lot of knowledge to share, and you give talks everywhere. But when a beekeeper and an online guy like me, I keep collecting things in one spot and another to put things together. It's always good to have a book, something centralized, organized, structured, and I thought that would be your book. The accumulation of all this information in a single structured way to give to beekeepers.
as much as we
How,
know now, yeah.
yeah. However, now that you mentioned that, it's more than that. I think it's even better because you're gonna go after the beekeepers and get personal, interactions and bring more information directly from the sources around the planet,
That's
which
what
is
I'm
great.
hoping, yeah.
Okay, okay. So it's more than that because people are always asking me the book is already done. Yeah, just because
A lot
like
of the research has
is the
been
book
done.
done yet? So like
So the
James
book is
just
not done yet. That's what the
yes,
Kickstarter is for.
yeah.
So we've got this to try and gain funding so that so it does the publication costs, and it does all the editing costs and the distribution and stuff like that. But also it's for a large part of it is going to be for the research and going to be to try and track down all of these cases. It'll take a lot of work. And it'll take a lot of time. So the book is set to release in 2026, around about May, and earlier if I can do it, of course. But that's the kind of goal we've set. And so we know of a lot of cases already, so we can reach out to and contact them. And of course, Marla and Stephen have a lot more experience than I do, and many more contacts, and well, which is why I'm going to be relying on their help. I'm the rookie in this scenario, so I've only been researching for about 11, 12 years maximum. They're the veterans, and I'm going to be counting a lot on their expertise to get this done. But I'll be the one running around and interviewing people and collecting this data and compiling most of it. So the Kickstarter I was a kind of a fun little platform, both to raise awareness for the book and also to garner support from beekeepers that could benefit from this. So we've got the book, which you can actually pre-purchase a copy on this Kickstarter, and we've got a few other really cool rewards as well. There's a teddy bear that I designed, and actually I do have it. I'm gonna show you. This is Melly, the honeybee, that I designed as a mascot for the project. So she's been turned into a little plush toy. So she's also available. We've got T-shirts, and we've got really cool posters of splash art, which I have actually done myself. So I'm a bit of an artist. So the book cover art and the splash art, this resist honeybee poster, they were both done by me. Which means you can also get signed copies, as well as signed copies of the book if you want to back the project. So we're really hoping for support, because this would just get this done so much faster and easier if we had a bit of funding to finish this up.
I'm sure a lot of people are gonna want a book like that, and I hope people at home support you. Let's go to the reward tiers a little bit one more time, just to make sure people understand which one is each. So which one is this one? The honey tier.
Yeah. So how Kickstarter works is you got the center
Yeah.
page, and then you've got this little sidebar on the right-hand side there that you can scroll down for all the rewards and stuff. So there's different tiers. The first tier is, pre-purchasing a copy of the book. You don't actually have to buy into a tier. You can just donate $5 or $10 if you want to support the project because you think it's a good idea. But the first tier is the honey tier, and that would be a copy of the book, a paperback copy of the book once it comes out. So if you make that pledge, then as soon as the book is published, you will be mailed a copy of that. I think the second tier is the pollen tier, and that gets you a copy of the book and also a poster of the campaign splash art, which is the honeybee against the floral background with the title Resist. So again, that was designed by me. And then I think the next tier over is you get both of those things, and then I think you also get Melly the honeybee, the plush toy that we designed for the project. And then you get into the higher tiers where you start getting signed copies of things. You get signed copies of the poster, or you get signed copies of the book. We do have 2 tiers that are designed specifically for beekeeping clubs. Those are the last 2 tiers. If you want to combine your backing donations, you can get Q&A with all 3 of the authors of the book. So an online Q&A that lasts for 1 hour that you can schedule with your beekeeping club. And we will come online and talk to you and answer your questions either about the book or about resistance breeding and stuff. So those tiers are limited. I think the very last tier is you can actually have you can have your beekeeping organization or your club or your own private name listed on a tribute page in the book that will be printed with every copy. So everybody knows you've backed the project and you were a significant part of the book. So yeah, those are we went through the rewards there. I think that's about it, really. We do have some stretch goals. So if we do actually surpass our needed goal, then we're actually going to do more. That's I think close to the bottom. So what we're going to do basically, if we have, I think one of the highest stretch goals is if we get over a certain amount of money, then I'll be making a YouTube mini series, hopefully where I interview some of these really cool beekeepers who have resistant bees and go through their processes live and in person, which would be really cool.
I can help with that.
Exactly. So that should be really fun. I hope we get there.
Yeah. Well, so people, I'm gonna make my purchase tonight, and I'm gonna back this up because I think that's gonna be a good book to have available for everybody. All right, Melissa, thank you for this overview. Let's see if we can get some questions here. Oh, we do. Oh boy, here you go. Am I missing something here? Here you go. That guy again. Oh, Lord.
Right. So we were talking about this. So the question is
breeding. Let me just
sorry.
Let me just read because I have listeners only. So is breeding the trait as simple as selecting for uncapping observations?
Yeah, so we talked a little bit about it in the presentation, and because we have this environmental variation, and because the different, the trigger levels are different at any given time, and they're unpredictable, it's not a great trait to be quantifying in order to select for resistance. Not unless you have some sort of thing that will trigger them into doing more hygienic behavior. Now there is something, a kind of a substance called UBO that is being developed. I think you covered. I think Dr. Wagner spoke about it on one of your older podcasts.
I covered many times the UBO.
That's being developed as something that can be used as a kind of a hygienic behavior stimulator, and that it may actually be a good trigger to start quantifying things like recapping. But unless we have some sort of trigger or a way to standardize the response level, it's going to be very difficult to make selection decisions based on the amount of recapping, because the amount of recapping is very dependent on the amount of parasite you have in your colonies, which is kind of a catch-22. If you have a lot of parasites, then maybe you don't want to be breeding from that colony. But if you don't have a lot of parasites, you might not see this behavior. So I definitely think presence is definitely a key into target colonies that maybe need a little bit more scrutiny when you're looking at it. So I'll talk a little bit about what I was doing after this paper. I was in charge of a breeding program in Hurdal, and I was in charge of designing a selection process and an algorithm that selected for the best queens. And what I wanted to do, because I wanted this method to be really easy and simple, I wanted to see if we could just select for low mite counts and breed queens off of that. And what I did was we used the mite counts on bottom boards 3 times a year to select for these queens, and then I was tracking the recapping throughout the population. And interestingly, when we just selected for colonies with just low mite counts at critical periods, like in August, just before winter, we actually did see a very large increase in recapping accuracy. So simply selecting for lower mite counts did actually push up the recapping accuracy and the hygienic behaviors we were tracking. So that was a very interesting point. And these colonies were free mated as well. So something with a little more drone control might actually have more of an effect and a faster effect as well.
Cool. Melissa, since you mentioned UBO, there is any article or some experiments correlating the two things and recapping behavior and UBO, or some information on that?
I wish I could speak more to that, honestly, but I haven't had a chance to talk to Dr. Wagner very recently, so I don't want to say anything that is incorrect or just
Oh, but
myself.
so that
But I'm
means
very
there is
interested in
something going
seeing.
on.
You'll have to look at her papers and what her lab has been
Ah,
putting out, because I
okay.
think it's very fascinating. I think we might have something there. I think it is still being developed. Don't quote me on that, but it the results seem to be improving. So
Kara Wagner, I'm gonna hunt you down. You're gonna come back to the show and we're gonna talk about this potential correlation with recapping and the UBO. Don't hide yourself.
I think you can actually purchase this to test, but I think
Yes, we can.
the cost
Well,
is
not in
still quite
Canada. I
high.
think
I
I
don't know
think
about
in
Canada.
Canada. I think Canada. I think she's in Canada now.
Okay.
I think you are able to get UBO in Canada now.
So I think the
And
limiting factor is probably the cost, because it is, it's a relatively costly to produce, but they're working on bringing that down.
oh yes, and yeah, I help Kara. Okay, I'm gonna make it public. Kara is one of the companies that I help in my consultant business, so I'm aware of some of the things going on. But yes, the price is gonna go down, and she's working hard on trying to. It's not easy to build a company from scratch, and she's doing everything herself. And she's a warrior, and I'm helping as much as I can, especially in this
Yeah.
industry that something happened, boom, everybody leaving, billions of dollars lost. It's just it's a very interesting industry too.
Very highly variable, and with
Yes,
with the
yes,
volatility of climate and the
yeah, yeah.
reducing habitat and stuff, we've got a lot of challenges to
Just
deal with
got
in
two
our
calls
environment.
yesterday from commercial guys that are giving up. They're just
Oh
selling
no.
everything and just trying to move to another direction now, and just ask me for help with
No,
what
don't
kind
give
of.
up. We could beat this. We just need we need a little more time.
They are in big debt and they just want to find a way to survive in a different way. They're tired. They're
Yeah,
done. Yeah,
I can
and
imagine. It's
if I
been really
got
tough,
two
especially
calls, yeah,
this year.
yeah. If I got two calls, and imagine how many people are right there that is really already giving up after this last episode of crap.
Yeah, no,
Okay.
I think we can probably put a little bit of thought into exactly how the operations are run in the large scale, collectively, and government regulations and stuff. Because there's things we could be doing to make it a lot easier for the honeybees,
Yeah.
and pollinators in general, in terms of habitat management practices and agricultural management and stuff. But that's probably a whole other podcast you want to do later.
Yeah, we can do lots of things later. I'm skeptical. Maybe I'm getting old in this business, and I saw a lot of players, and when things hit the media, it's so hard to find the truth,
Yeah.
and it's kind of complicated. Well,
Yeah. Well,
anyways,
it's good
this
having
is
scientists on your podcast, because a lot of them are
yeah.
kind of up close and personal with the actual data. They're pulling stuff in from the field, so they see firsthand what's going on.
Yeah, but they have their biases too and
They
their
do.
agendas too.
They're human.
Yeah, they're human, yeah. And I know them better than
So
better than the others.
I'm trying to be as neutral as I can and
I
just
know.
working for.
I don't wanna you in trouble. I put myself in trouble. A lot of them have agendas, and it's not good.
But I have to say, I've learned a lot from the beekeepers I've worked with. Honestly, Tarja
Good.
Reinhardsen, I worked very closely with him during my
Listen to the
PhD, and
beekeepers. Yes.
we would sit down every morning before we went to work, and when we would just, have conversation over coffee about bees, about what they might be doing, about observations that he found, and we just talk over breakfast, and then he would go to his bees, and I would go to my lab, and that's how it worked day to day in Norway. But I feel like I owe him a lot. So we spoke about this book. Like over a year ago. This was Tari unfortunately passed away a year ago, November. So he never really got to see this completed, but we spoke about doing a book. And that's kind of part of the inspiration also why I want to get this done so badly is, I said I would do this, and that's, one of the last things that we spoke about. So I want to make this a reality,
Good.
for him and his memory as well.
Good.
He
I'm
was a
glad you're
huge
going
influence on my work.
to that path. Listen to the beekeepers. Keep an open mind. Okay, let's get more questions here. All right. Jason, what's the best way to find uncapping, recapping behavior?
That's a cool question. So once you've trained your eye, you can actually see it from the tops of the combs. But while you're learning to see it, how I did it when I started was I very carefully kind of flipped open the caps and looked on the underside. Because what you see when a cell has been uncapped is when the larva pupates, it leaves kind of this silk cocoon around the entire interior of the cell. And if the cell has been broken into by worker bees, the underside of the cap is actually going to have a hole, and the wax they use to fill it up again is usually darker. So if you want to really get in and look for this recapping behavior, because that generally holes are sealed, so you can get lucky, you can pull out a frame and see the holes in the brood, in the the brood frames, and you will see that with a recapping colony. But if you want to get kind of an idea of the frequency of how much they're doing it, you can flip open these caps and you can see which caps have been tampered with and which caps haven't. So it does take a bit of training, but you can definitely see it with your eyes. You don't need a microscope or anything to check this. Sometimes the caps are completely torn off and then just replaced with wax that just breaks apart when you put your hive tool on it. So just like explore some of the frames,
Just
and see it. You'll find it.
just to add a little thing for my South America friends, this is different than the halo of propolis that Africanized bees put in each cell. In Africanized bees, when you
Oh,
open the cap in a board of each cell, there is a line of propolis there, and
that's fascinating.
it is different than the recapping thing. It's not all Africanized, but there are a lot of the Africanized. Sometimes when there is a lot of propolis in the surroundings, there is this little line. Nobody really know why. That they just put the line there and then they close. Is quite cool. Maybe I should bring some of this material for the American North American people to see some different things.
Yeah.
Okay, next. Oh, this is cool. Let's be frank. Can we add water inside hives to raise humidity to fight mites? Treatment free, right?
I mean, I think
That's
we've some people have definitely
that's
thought of that
interesting.
before, but I like to. I'll say one thing about the honeybees is they are so obsessively good at keeping the colony just how they like it, that they'd probably correct the humidity difference too quickly to affect the mites. They're really on the
Yep.
ball with that.
That's the come back to what I was saying. The bees are really good to keep the environment the way they want. If you have a hive that have perfect isolation, they almost need almost anything. They don't need almost anything. Is if isolation, just like in a tree, in a small hole, when you look with cameras and do the measurements, it's so constant. The amount of CO2, the everything there is so perfectly designed that they control everything. When they need more, it's just like a lung. They kind of breathe. It's beautiful.
Yeah, I will
So
say that there are some people working with wire embedded frames that actually heat up, and they heat the brood not high enough to kill the brood, but enough to interfere with the mite reproductive success. And although this method has actually had some success, but putting wireframes in your colony, it's a little costly. So again, we've got a
Yeah,
lot of things that work, but getting them to the point where they're easy to use, fast to use, and cost effective, those are our big problems here.
yeah. I love innovation. I want to see the results of these things, but sounds to me that when you take the decisions from the bees, it's just like the government taking decisions from us. When we, it is never good, never good.
I mean, it was the principle of most of the beekeepers I work with. It's this let the bees lead. And
Yes.
they tend to be able to overcome pretty impressive things.
Okay.
So, yeah.
Let's go to the next one. Connor, is there a risk of selecting for tolerance for Varroa and associated viruses rather than actual Varroa resistance, VSH, SMR, by selection through not treating as opposed to measuring VSH, SMR traits? Interesting.
I think there's definitely a level of tolerance that comes with this type of breeding. Um, but a risk of tolerance over resistance, I haven't seen it yet. I think most of the populations we covered, they either have a combination of both or they're predominantly resistant. Most of the traits that I've observed in different populations seem to be directed towards controlling the mite levels. But it's possible you could get some levels of viral tolerance in it as well. Viruses tend to evolve much more quickly than parasites or honeybees, so it might be a little harder to retain that type of tolerance in populations. But I mean, if you select for natural traits, what you're gonna get is a handful of traits that are probably best suited to the environment that you live and work in, and that your bees are used to as well, which is why I always promote performance-based selection rather than trait-based, because what you're doing is you're selecting for the traits that work for you in your area, and it might not necessarily be the same as what works in a different area. So you have a lot of scientific evidence, but the evidence is all collected from one particular group of bees that may not necessarily have the same environment as your bees. Does that answer the question?
Well, that's another good question. I still thinking here, and I understand what you're saying, but I don't know if I have an answer for that. I'm gonna sleep on that one and maybe make a video about it. Interesting. Yeah, there is a chance, but well, let me think more about that.
We did some viral work in Ontario's buckfast stock, and we found that actually it was a reduced prevalence and quantity of viruses, usually linked to the reduction of the mite.
Okay, send it to me. That will become a video too. I like those
Okay.
topics. Okay, Connor again. Connor, thank for the question. Oh, another Nina Sokolov's 2025 paper on tragedies of parasites tolerance in Darwinian beekeeping. Is this
Ooh, that's a
related with
new
your
paper.
question?
I have not read that one yet, but
I
now
would,
I'm gonna
I'll,
put it on my list.
yeah, I'll check that out. Okay, so there is information. So I'm not gonna open my mouth until I read this. Okay, good.
I think I'm agreed. I want to comment on it, but I don't know what it's about, so I'm gonna
Yeah.
read it first.
All right.
We got one from Let's Be Frank, I think. Oh, let's do this one first.
What guidelines can you set up for small scale operators or even larger to start pursuing resistant breeding in their apiaries?
All right. So we haven't written the book yet, so I don't have all of the information that I want to have to be able to make these recommendations. But from my limited experience and the experience of the beekeepers I worked with, what seems to really work is if you take a small subset of your colonies, don't take all of them and do it all with them, because if you decide to stop treatments cold turkey, you will lose a lot of bees. What you can do is you can do it progressively and you can do it collectively as a group. So each of you, if you have a community of, let's say, 10 beekeepers, you can each kind of put 10 into monitoring program, a population where you slowly reduce treatments. And you use a kind of integrative pest management where you're treating only when necessary and you're counting mites. And then you can pool all this data and select from those 100 colonies, if there are 10 of you, the best queens. And then breed from those and replace your stock. So effectively, that's what we did in the breeding program I was in charge of for the last 4 years, 5 years in Norway. Is we had a group of beekeepers that joined this project, and we had a queen breeder who was supplying the queens for these, and I would monitor all of the 100 plus hives that we had, based on the data that the beekeepers gave me and the data that I collected on the mites. And I fed all that into kind of an algorithm that I designed, and we had a point system where we would score the queens based on how gentle they were, how much honey they produced, how healthy they were, and their mite counts. So I used Taria's bees and their mite populations as a baseline to what we wanted to select for. So any bee colonies that had higher than 10 mites a day in August, which was the critical period just before winter, they actually lost points. We wanted them out. We didn't want them in the study at all. And anything that had 10 mites or below, preferably lower, they would start getting scored for real. And then we would take ones that had the highest honey production and were the most gentle. Anything that scored less than perfect on gentle, we also took out, because we wanted nice bees. These were commercial bees. And that's how we actually started to advance the increased levels of recapping, and the brood infestation rate did drop in the final year as well. It actually tanked, so that was really good. So that'll be a paper coming out. But yeah, to sum up, the recommendations I say is work together. Don't use all your hives in your experiments. Use some of them. And I would say select on low mite counts, and then just be aware of your surroundings, be aware of how many colonies are around your bees, what's the status and health of them. And if you see one of your colonies really getting out of hand with mites, then don't hesitate to treat it and just remove it from the pool. Just don't let it breed with the bees that you want to breed from. I think in Herdal, I recommended putting queen excluders underneath the hive to prevent drones from getting out. It's an easy way to just reduce the number of drones from the hives that you don't want breeding with your stock. So yeah, I would say slow and steady, integrative pest management, and try not to kill your bees. So do treat and just remove that queen from your breeding population.
Cool. Yeah. You got your guideline, James. Connor, also, is there a risk of losing important rate traits and alleles by breeding for specific traits like VSH? And how can we compensate for that? Referencing Jack von Alphen's work.
Yeah, there's a lot of opinions on that. Is how to keep your how to keep your population from losing traits. So that's why we incorporated desirable traits in the stock when we were doing this breeding program in Norway. But we also had kind of an open mating system as well, so we didn't limit the drones and the drones access to queens. And we did this because the genetic diversity in your population is incredibly important. And this is one of the ways you're going to keep beneficial traits in your hives. So a lot of what breeding has been historically is really controlling the traits, controlling what gets to breed and what doesn't. But that's not really how bees operate. They have the queen mates with a number of different drones, and that's natural. The queen also, the drones compete for queen mating. That's also natural. So if you're doing something like artificial insemination, or you're limiting the number of hives that have access to the queen, what you're doing is you're limiting the natural genetic variation traits of the bees. As of yet, we don't really know exactly. It's difficult to quantify the level of effect that this has, and how quickly you might lose beneficial traits versus not. But I would say, if a species has had over 160 million years to adapt and evolve, the system that they have in place now is probably going to be one of the better ones for them. So maybe trying to work with that system instead of controlling it might net you better results.
Cool. Let me just add something here because I don't know if you guys follow me in my newsletter, but if you don't, the University of Montana beekeeping courses, they have a very good courses, and they're going to give us some of those courses for free to my audience. So if you're part of my newsletter, and why I'm saying this is because they have a very cool system that in that course that I was able to see to to control those drones and keep those alleles in one place. They have a very cool system. I cannot talk too much because I want the guy to teach himself how to do it. I'm bringing the professor here, so it's pretty cool. But so if you want to follow this along, or maybe to get this one of those free courses, uh, please sign up to my newsletter. It's free. So yeah, it's cool. It's coming. So what else? Okay.
There was a question from Let's Be Frank. There you go.
Yes. Thus, adding light, UV, etc., inside the hive hinders might simulate open hives like in warmer countries. Let's be frank. I think, yeah, I know where you're going with the point, like, because I mentioned many times that the mites are very sensitive. So, however, therefore, if we do something inside the hive, might affect them. The logic is there. Is the logic is spot on. The problem is the bees are so good. They to defend their brood and to keep the system so tight and protected from everything that is hard. If you push too hard, meaning is gonna that means you're gonna start to hurt the bees, and then what's the benefit? You're gonna need to assess yourself. I don't know any light of UV that have any impact that I know of, but Melissa, do you know anything about it?
No, I don't actually, because I haven't come across any papers that have studied this in depth. I would guess though that we know what light does to colonies. They generally don't really like a lot of light in their hives. We know
Yeah.
observation hives tend not to do very well for prolonged periods of time. So I would say maybe putting UV light, because it is in their visual spectrum. They can definitely see it. You'll light up their colonies. It might actually do a bit more harm than good if you stress the bees via just shining lights. Like imagine if you had a home and you turn the lights off at night to go to sleep, and then someone just puts a sports stadium floodlight into your apartment. I don't know. That would stress me out.
Yeah,
So
well,
it's a speculation. So I
yeah,
don't know.
speculation. Yeah, maybe
I do know that the
try.
mites are trapped inside the cells most often, so they do have some sort of a barrier to UV light that the workers wouldn't have. So they'd be exposed to the direct UV light. I'm not sure. Ultimately, I don't know.
All right. Hello, Castle Hives. Hello back. Okay. Let me see what we got here. Do you see any other questions, Melissa? I'm scrolling here.
We got one by Lon Woodard, I see there, but I might need to ask for a bit of clarification.
Just hey, Mike. Yes, this is a bottleneck. We're working on that. Kara is working hard to find a way to lower the price, but life
I
is
haven't had a
hard.
chance to test out the UBO myself. Like I want to get my
Oh,
hands
you're
on
gonna
some and
like it.
see if it works.
It's fun. Actually, it's fun. It's not only the job itself, but it's fun. And for me, it have more emotional involved because I was in my postdoc in Dr. Olof's Ruppel lab when Chiara was doing her PhD. So I follow her
Okay.
struggle for years. Oh boy, and then come up with something new, bring something new to the world. It's just proud of her, and I know how hard she worked. And so I have this emotional connection with this project because I know how hard she worked to put that, to bring that to all of us. Okay, information here. Dancing Bees has UBO in Canada. I'm assuming is UBO. So thanks,
Oh, very
James.
cool.
I'm doing propaganda for you, Kara. By the way, there is no.
So we can maybe do there's a one from Let's Be Frank that was further up, 8:02 p.m. I can read it just if you want to. 8:02.
8:02 p.m.
There, yeah. The question is, can we get a list of things that failed to work so we don't make the same mistakes?
Let's Be Frank
Um,
is smart. Yes, that's the.
things that failed to work. I do know that Tadia did agree to export some of his queens to various areas, and again, putting them in environments that they weren't generally suited to did kind of reduce the resistance rate. So I would say importing is probably not where you want to go if the import site is from far away. I would say staying local. In terms of what else didn't work, I can't speak a lot to that because I worked with beekeepers that had already done it. So I was retroactively looking at the mechanisms that worked because they did work, and why they were there. So what didn't work? I'm not sure. Perhaps actually some of the beekeepers in the audience could be better suited to answer that type of question. So I will
Yeah,
say I'm not a beekeeper. I have worked with bees, I've cared for bees, but I can't really claim the title of beekeeper because that's I just research them. I work with beekeepers.
yep. If somebody have any information, please write to the chat so people can know. I can put it here. But let's be frank. Let me add some things on that. Sometimes you, I know the logic is correct. You don't want to do the same thing that somebody else failed. However, sometimes because in your environment, that something that was bad for somebody might work for you. I just want to keep that in mind that the logic with bees is not that simple sometimes.
Actually, you made me think of something there that I
Okay.
can add. So when I was looking at the Norwegian colonies, I found a lot of recapping. I didn't find a lot of brood removal. Right. Well, Stephen Martin has done extensive research on resistant traits and stuff, and he actually found that brood removal is kind of the dominant trait in a lot of different populations. But he works with a lot of populations that are much further south. So you're exactly right in saying that environment does dictate. So if you think about a very far north temperate climate, they don't have a long summer. Then and winter bees are much more important. If they lose those, then they lose the hive because they need to survive until the snow melts. So it could be that recapping is the fallback to mitigate brood removal, so they're not losing a lot of these winter bees. Whereas in a more southern climate, brood removal might be more highly favored because it's easier to replace the brood because they have access to more resources for longer. So yeah, you really need to think about your environment and the challenges that your bees face in that specific environment, and then think about the types of traits that could be beneficial and the types of traits they might evolve.
Got it. Okay. Connor is saying things to you, Melissa.
Thank you. Now that's an interesting comment at the bottom there. Bruce Southern Sass apiaries. I
Okay.
have something to say to that. So if you're catching the drones when they return from their flights, that might be a selection bias in itself, because if you're catching the drones that return, it means they weren't successful in their mating flights. The drones that are successful catch the queens and they die, so they don't come back.
Good point. Yeah, that's a good point. The ones that come back failed. Cool. All right, Melissa, I think we provide a lot of information for the folks that were asking for information. Wait, Mike. I saw it. That question. Any thoughts on possible missing enzymes in mite-resistant bees in Norway? There was one mention of the mites trying to feed and starving. It's from a
I'm
specific publication that you saw, Mike. I
I'm
don't
not
know what you're
sure
talking
how to
about.
speak to that. No, I haven't heard of mites trying to feed and starving or missing enzymes. I can say that we haven't done a lot of proteomics with the Norwegian bees. So we haven't done a lot of molecular stuff with the Norwegian bees. Mostly I've been working with beekeepers and tracking behaviors and trying to find tools that benefit beekeepers directly. I would love to start doing some molecular stuff on them, but it wasn't in the grant proposals that I had working on these and researching the bees. So
Yeah.
I don't think I can answer that question.
So Mike said it was a talk. A talk and haven't yet find it yet again. So no problem, Mike. So just to add to that, a little secret, Melissa, I want to tell you. I learn a lot from my people here. A lot
Yep.
of the things they been sometimes they bring something that I had no idea is happening. That's why I like to be in the internet because and have the audience because we work like a social community kind of thing. I learn a
Yep.
lot from people bring me something that a lot of things that sometimes I have no idea what we're talking about. So let me check it out.
Yeah, no, it's good. And then I always get tipped off from various articles from people and things that have been in the news and stuff. So it's a good network.
Yes, it is. Yes, indeed. Okay, I think we reached the end of this. Melissa, thank you very much for your time. And all the work you put to all of us, all the information and this new project with the book. I hope the people at home will support you as
Oh
much
yeah,
as they
spread it
can.
around. Do share that Kickstarter. We need as
Yes.
much support as we can get.
Oh, let me put the
And I wanted to thank you, Humberto, very much for having me on your show again. This is always fun.
my pleasure. I want to put this here. Boom. Here we go. So now everybody can see the Kickstarter. If you want to support this project, I think it's going to be a new book in everybody's shelves and with good information. So one more time, Melissa, thank you for all the work you're doing. I think the beekeepers appreciate your work. I certainly appreciate your work. And if you have any message to anybody, Inside the Hive.tv is open to you anytime, anywhere.
Very nice. Cool. Yeah, no, keep going, guys. Don't give up. I know beekeeping's hard at the moment, but bees are incredibly resilient. They're spectacular, and if they can do it, so can we.
Yes. All right. Let's keep together, my friends. Good to see everybody here. It's a couple of news. I am going to a direction where I'm going to do more live streams soon regarding teaching bee biology live. If you guys have recommendations of things that you want to learn about bee biology, I think I'm considering to have a chat per week, like we can get together and talk about bees and I can teach bee biology in more details and take questions from people. And yes, you bring your beer, whatever you want to drink, and it's just let's have some fun learning about those things. So if you have recommendations, please send me to my email contact at insidethehive.tv and I'll do the preparations from those seminars and we're going to have fun together. All right. So Melissa, thank you again.
Thank you very much.
And for you guys at home, thank you for joining us today. And I'll see you guys in the next one. Inside the Hive.tv, the show about bees. See you guys in the next one.